Hello everyone. In this video I want to go over DNA replication. And so if you haven't learned about the structure of DNA, if you really don't know what that is, you may want to go back and look into that, get an idea of how DNA is put together, what it's made up of. That way you really know what we're talking about in this video. So let's dive right into it. What is DNA replication? Really, the definition of this is just copying DNA. It's the process where DNA is copied. To replicate something is to make a copy of it, and so DNA replication is the process that happens in cells where the DNA gets copied. And we'll go into why that's useful in this video. We're going to talk about how it happens. It's a complex process that actually involves several different enzymes and different molecules. In this video, I just want to give you a basic overview. So we're only going to talk about two of the enzymes in this one. A little bit about maybe we throw in a third one just for good measure. But we'll talk about those enzymes and just give you a basic view of this. So let's just take a look at how this happens before we really talk about why this is important and really what's going on in detail. In this picture, I didn't put the word on this slide here, but I do want you to know the term semi-conservative model. This is what we call a semi-conservative replication model. So let's break that word apart really quick. Conservative or conserve means to save, and then semi-conservative means we're saving part of it. And so really what we look at here is we have our parent strand, our original DNA strand, and we're going to save part of that, and then we're going to have a new part later added to that. I'll show you what that means in this image here. So this is what we call our parent strand of DNA. This is the original. This is the one that's going to be copied. We have two complementary sides based on the base pairing rules. So A and T go together, C and G go together, or adenine and thymine, cytosine and guanine. So we start with this. In order to copy this, though, we need to open it up. And so this is one of the first enzymes we're going to learn about here, DNA helicase. DNA helicase opens up this DNA strand. It breaks the hydrogen bonds between the nitrogen bases. And so the helicase breaks these hydrogen bonds open here and exposes the nitrogen bases on our parent strand. Our second enzyme, DNA polymerase, is going to come in and it's going to add new nucleotides according to the base pairing rules here. So if there's an adenine exposed, it's going to add a thymine. If there's a thymine exposed, it's going to add an adenine, and so forth and so on all the way down. And because it's following these base pairing rules, we end up with two identical strands here. This strand should be the exact same as this old parent strand here because they match up, they pair up according to those nitrogen bases. At the end, what we have is two daughter strands. So we had our parent to begin with, and we've created two daughters here. But we have a semi-conservative model. So in each new daughter strand, we have one side of it is coming from that parent strand, and then one side of it, one of the new sides or one of the strands here is all new DNA. And so this is what we mean when we say semi-conservative model. We have one old and one new strand, one new and one old strand. And that's the way this works. Why is this important? Well, when cells divide, each of the new cells that are formed need to have the exact same copy of DNA inside them. Let's look at it this way. If I burn my hand, I've damaged some tissue, I've killed some cells. Well, the cells that are going to replace that are going to come from the living cells that are around that damaged tissue. As those cells divide and replace the damaged ones, I want the new cells to be an exact copy of the ones that were damaged. And so to do that, to make that happen, the DNA has to be copied. If you've learned about the cell cycle, this happens during interphase in what's called the synthesis stage of the cell cycle. And that's when DNA replication takes place. If you haven't learned about the cell cycle yet, you'll get there. And this is what's going on during that stage. So you'll already be ahead of the game when you learn that. But so that each new cell that's formed has the same instructions or the same blueprints, the same DNA, the same genetic information, the DNA has to copy itself. And we want an identical copy so that each new cell can do its job correctly, can make the right proteins and do all of the processes that it needs to do. Here's just some interesting information about how quickly that DNA replication process can happen. So let's look at how we can copy 6 billion base pairs in just a few hours. I mean, that's crazy to me. I can't even count to 6 billion in my lifetime, much less copy something that's got 6 billion parts to it in just a few hours. And so how does this happen? Well, if we look at DNA as this big long string of genetic information, which really it is, each chromosome is one long strand of DNA. DNA replication is going to happen at several different places on that molecule. And so our helicase molecule is actually we're going to have two helicase molecules going in opposite directions here. And we'll have the same thing happen down at this point on our DNA strand. And then further on down, we'll have it again, and so forth and so on, all the way down to the end of our chromosome. These are what we call replication bubbles. And so we've got replication happening at both places, going in two different directions. And that's how we can copy this very large molecule in really a very short amount of time. And so ultimately, we end up with two different DNA molecules there. So let's look at how this takes place, how this happens, how these replication bubbles get to move on down the DNA line, and what's really going on there. And so what I want to do now is actually take this bubble, and I just want to focus on one little side of it. So we're only going to look at this one part here. Just know that the same thing's going on here, and here, and here. So let's take a look at this in more detail, zoom in on that, and see what's happening. So here's what we call a replication fork. This is one side of our replication bubble. We said earlier that our helicase is moving down and it's unwinding, it's untwisting the DNA helix, and it's unzipping that DNA molecule so it can expose those nitrogen bases in the parent strand. So our helicase moves in this direction and breaks open our DNA molecule. And then coming right behind that, we have our DNA polymerase, which is going to read our parent strand and add nucleotides to those exposed nitrogen bases. So here this polymerase is going to add a nucleotide that has a guanine on it, and a nucleotide with thymine and cytosine and adenine and so forth all the way down. Up here at the top, you've kind of got a definition of what each one of those two things does. And so this is how this happens. It's just moving along, copying that DNA. What I want you to see in this picture, though, and this is what I want to go into some detail about, this polymerase has an arrow showing that it's going this direction. Well, this polymerase here is going in that direction. And so what we see is that DNA replication is actually happening in two different directions. This has everything to do with the fact that DNA is what we call anti-parallel. So if you don't understand the anti-parallel nature of DNA, please go back and learn about that, because this next part will just be confusing to you. So here's why this happens. Let's look at that in more detail. Again, let's zoom in on a replication fork here. This is a little more detailed picture, and if you look at the picture here, you can see that there's a lot of molecules that I don't really want to go into in this video. I'll do it in a later, more detailed video here. But here we have DNA. Our parent strand is the dark blue. Our new strand is the light blue. So what I want you to recognize is that, remember, DNA is anti-parallel. And on this end of our parent strand, we have what we refer to as the 3' end of these nucleotides. And so this is the 3' end, and if I follow this down, moving from 3' all the way down, I get to my 5' end. And so that's, again, that anti-parallel part. If you don't recognize that, if you don't understand that, please go back and look at the structure of DNA and why we call these 3 and 5'. But being anti-parallel, this side is 3', and so we follow this down all the way, and we have our 5' here. So each side is going in different directions. Well, when we learned about DNA polymerase, as we started to figure out how this molecule works, we realized that it's moving down the parent strand in that 3' to 5' direction. It's building our new strand in a 5' to 3' direction. And so this is the direction that DNA polymerase moves. Well, if our helicase is going that way, that's great. This polymerase moves this way. If we look up here, this is what we call our leading strand of DNA. And we call it that because it's just following that DNA helicase, and it's doing this continuously. It moves pretty quickly, and it can do this uninterrupted. Our bottom strand here is what we call our lagging strand. And you guys might recognize lagging as like a video or a video game. And when it lags, it just kind of starts and stops and starts and stops and skips that way. Well, that's exactly what's happening with our lagging strand here. Because this is a different direction than our leading strand, remember, we have 3' going to 5', this is the way our polymerase wants to read our parent strand. And it wants to build our new strand going from 5' to 3'. And so it's going to do this in sections because our helicase is going this direction. So what happens is polymerase will build here, and then it goes back and catches up with helicase and builds, and catches up with helicase and builds, and comes back and catches up and builds again. It builds it in fragments. And these are what we call Okazaki fragments. The Okazaki fragments are those sections of DNA that are replicated on our lagging strand. And that really happens because DNA is anti-parallel. That third enzyme that I said we might get to is DNA ligase, and its job really is to put these fragments together. So this is the result of DNA being anti-parallel. I want you to recognize our leading and lagging strands, and the reason those are that way is just mainly because the DNA polymerase reads our parent strand from 3' to 5' and builds our new strand from 5' to 3'. I hope that clears that up a little bit. I hope that kind of makes some sense. And I will put together a more detailed video of this later on.